You can always ask, but I am Ethan Perlstein. My colleague Whitney Dolan and I are Per-Lara Cure Guides that are acting as the Project AP research team, a distributed research team. I'm based in Oakland. Whitney's based in East Coast in DC. And so we're excited to present this series of monthly forums where we invite researchers that are going to be working on Project AP to talk about what their plans are and to have the possibility for open conversation with community members, with family members. We also have other scientists who are researchers themselves working on a different, their own project who will be available on this call to chime in as well if they feel like it. We really want to have this be an environment where we can have a scientific conversation, where we can try to make it as accessible as possible, but we want to try not to dumb things down. We really want to try to tell you what's going on and give you a chance to engage. And we have a little bit of a time at the end of the call for those who can hang out, especially if you wanted to get more clarification on points. There'll be an opportunity for that in a little overflow session. So I want to just give a brief update that on the research side, we're excited to have with us today several researchers that we're going to be working with, that Project AP is going to be providing funding support to. And I'll have those folks sort of introduce themselves when we do our rapid intros. And I want to also sort of comment that we are working behind the scenes on a lot of research infrastructure. A lot of capacity building has already happened, and I think Bina has communicated that. But we're doing other things that are behind the scenes, including stuff like getting ready to reprogram peripheral blood mononucleoside cells, PBMCs, and get them into iPSCs. And that will allow us to do all kinds of interesting downstream work. So those wheels are starting to turn, and we're hopefully going to look at around 10 to a dozen lines at first. We're not going to be able to process and reprogram all the available patient or AP hero lines, but that will, we have got to get started somewhere. So that process is starting to move, and we've also now kind of consolidated and kind of reconciled various lists to create a biobank that really captures all of the various samples that have been acquired, and kind of reconciles, as I said, multiple lists that have existed before, and really trying to make sure that information is available to anyone so researchers can follow up on that angle. So there's lots of stuff that's been going on behind the scenes in the last month since we first convened. So I want to just maybe do a quick round of intros because we have a slightly different attendance from last time. We have, again, a diverse audience here of families and researchers, folks who are more or less technically savvy. So we want to just try to, again, maintain a lab meeting type of ethos, which I think the researchers will appreciate. But we also want to have all the families who are listening feel free to chime in or wait or use the chat or use other means to communicate if they so choose, or just listen. So quick intros. I don't know if everybody sees the same array that I see in terms of who's tiled where, but I think maybe will be super helpful is I can call out maybe some of the researchers first, and they can kind of just sort of say, who they are, where they're located, and sort of just roughly what they're going to be kind of working on. I'll start with Nicoleta, who will be our presenter today. Thank you, Professor, for joining us. Maybe you can get us started here with this, maybe let's say 10 seconds or so of introduction.
Hello, thank you for inviting me to this meeting, and thank you for having me in your group. I'm Nicoleta Moisoi. I'm Associate Professor in Pharmacology. I am living in Leicester, and I'm working at the Montfort University in United Kingdom. And our work on this project is going to focus on characterization of mitochondrial function and dysfunction, and also on DNA damage and repair in HP. Thank you.
Thank you, Nicoleta. Let's see. We have also Professor Aryeh. Would you introduce yourself?
Hi, I'm Aryeh Warmflash. I'm an associate professor at Rice University in biosciences. My lab studies early human development using human embryonic stem cells, and in particular, we create models in vitro that mimic some of the aspects of what happens in the human embryo And our hope is to use those models together with reprogrammed lines from 8P patients, as well as with knockouts for particular genes to try to evaluate the biological implications of those different genotypes.
Thank you. Professor Hiroi.
Hi, thank you for the invitation, and it's wonderful to be here. My name is Hiroi. I'm an assistant professor at the University of San Diego, California. Where we're located in San Diego. My group focuses on utilizing stem cells and mouse models to understand neurodevelopment. We're predominantly interested in the genomics and how the 3D genome folds and alters 3D genome architecture, as well as transcription as a consequence of genomic imbalance. We study Down syndrome and other neurodevelopmental disorders.
Thank you. And let's see, Professor Stefan. Oh, you're muted.
Apologies. My name is Stefan. I'm at the University of Connecticut Health Center, the medical school there. I got interested in neurodevelopmental disorders through an X-linked copy number disorder, results in an intellectual disability, and our work on trisomy 21, human iPS cell models of trisomy 21 and an inducible gene dosage correction to study exactly how it results in neurodevelopmental phenotypes and which of these phenotypes can be reversed.
Thank you. And we have some other researchers joining us as well. Glennis.
Hi, everyone. I'm a postdoc at the University of Washington in the Department of Genome Sciences here in Seattle, Washington. I do long-read DNA sequencing and assembly of human genomes, and I was first to complete the complete sequence of chromosome 8, which is why I'm part of this project. And so I've been working with Bina and others in the Project 8P Foundation to basically sequence and assemble genomes from trios, so parents and their child, in order to develop patient-based reference genomes that can help us to detect and treat diseases in the future.
Thank you. And then Joe is joining us from Rarebase.
Hi, I'm Joe Bellucci, the Director of Scientific Partnerships at Rarebase, which, as many of you know, is a startup based in Palo Alto that's focused on making patient-derived cell models and repurposing FDA-approved drugs for a variety of different disorders in the rare disease space.
Thanks, Joe. And I'm still learning all the names and faces of family members. So maybe Bina, for this call, do you mind just kind of running through the tiles here and making sure everybody is covered?
Sure. I think for the families, it'll be easiest is if you guys just put in the chat your name or rename yourself with your 8P heroes, you know, kind of sub-diagnosis, deletion, or inversion duplicate. That would be helpful. And then I am Bina, which I think all of you know, and helped launch Project 8P a few years ago. And so, again, we're just really excited to kick off these roundtables, frankly, in the spirit of team science. And, you know, it's very much an open meeting. So feel free to put your questions in the chat, and we'll try to stay on a little bit longer too. And if they're very specific to a specific researcher, or the presenter today, which will be Nicoleta and her team, we can also help facilitate those conversations directly with the researchers. So again, please don't be shy, but if there are questions you don't even know how to phrase, we are here to help you, Whitney and Ethan as well.
So yeah, so that's a great idea. Please use the chat. If you're not able to use the chat and you want to just say a quick hello, you can definitely go off mute for a sec here. But yeah, I think maybe in the interest of time, if everyone can just do kind of as Faye has started here, that would be awesome. Okay, perfect. So we've got that going. So please, I'll remind our researchers to take a look at that chat here so that they can start to connect the hero with a face and a family. So with that, I want to just do a little bit of introduction for Nicoleta here to kind of set the stage. So as I kind of presented before, Project 8P is over the next 2 years really going to be pursuing multiple tracks of project support. And 2 of the tracks that are kind of already in motion include a drug repurposing repurposing track and a neurological disease modeling track. And so we'll hear more about the disease modeling tracks next month and in other sessions. Joe and RareBase are pursuing a repurposing strategy that's focused on iPSC neurons, and we're really excited about new, I think, data readouts that are going to hopefully be coming soon. But we're also thinking about other drug repurposing strategies, and especially strategies that could take advantage of cells that are much simpler to grow, like fibroblasts, and also cells that potentially they're simple, of course, and skin doesn't necessarily really embody all the aspects of AP, especially neurological symptoms, but we think there are aspects of what is called aneuploidy. So when a cell has an imbalance of its chromosomes, whether it's AP or other chromosomes that are out of whack, there could be certain cell stresses that are activated. And could we exploit simpler cells that are take less time to develop and are more limited, certainly in ways, but are kind of this minimum viable approach? Can we exploit those simple cells to look for phenotypes, in this case related to mitochondria and other cell functions that might be going awry in AP cells? Because we've seen this happen in other cases of cells that are experiencing aneuploidy. So we're excited to pursue this track with Professor Moisoi, and again, we're going to be taking a diversified approach to all the research that we're doing, but we want to really highlight projects that are in motion. And so, Nicoleta, I'll pass it over to you. I think you should have screen sharing abilities. Again, be mindful that this is an audience of peers and scientists and other AP researchers or nascent researchers, and obviously families. So it's kind of a mixed crowd, but I definitely don't want you to feel like you have to dilute down the science too much. We definitely want this to have a lab meeting kind of feel, so that if for some of the families out there, if this is a little bit over your head, or some of the terms are a little bit need clarifications, it's okay. We totally expect that. If you're bold enough, you can definitely use the chat, and Whitney and I will be monitoring the chat to try to answer any questions we can to make this feel like not just a one-way conversation, but also trying to keep in mind that we've got different levels of expertise in the audience. So with that, Nicoleta, please take it away.
Thank you very much for your introduction, and thank you for having me joining your group. And thank you, Bina, for getting in touch and interacting with me. And hopefully my work and the work of my group will be helpful for the achievements that we want to get in this work. So what I'm going to try to do today is to give you an overview about my background work and how our expertise can help the project. And for that, I'm going to give you an overview in the mitochondrial homeostasis and stress signaling mechanisms and how they contribute to neurological and neurodegenerative etiologies. And for that, I'm going to focus on mitochondrial dysfunction during neurodegeneration and neurological disease. And I'm also going to touch base on how mitochondria communicates with other parts of the cell, particularly the mitochondria nucleus communication, which is very important in designing stress and quality control signaling mechanisms. And from this, we are going to look at how DNA damage and repair mechanisms may influence, going backwards, the mitochondrial homeostasis in neurodegeneration and neurological pathologies. And I will also show you what is the project plan coming out of this background information. So some of the key defects that have been identified during neurodegeneration and in neurological pathologies that are triggered by genetic susceptibility, environmental factors, lifestyle, or aging include mitochondrial dysfunction, DNA damage accumulation, protein aggregation, and also impaired lysosomal function and trafficking. So these hallmarks are the result of molecular and cellular homeostasis that includes oxidative stress enhancement, lower ability of the cells to produce energy, metabolic perturbation, and also imbalance in the ions that are processing the signaling in the cells. All these things lead to neuronal dysfunction and cell death in these diseases. So how mitochondrial dysfunction appears in neurodegeneration and neurological disease? So let me just show you and give you a background about mitochondria. So mitochondria is a part of the cell, an organelle, as we call it, which has over 1,000 proteins that are encoded by the nucleus. So the nucleus is designing how these proteins are made. They are produced in the cytoplasm and imported in the mitochondria. A few proteins are also produced by the mitochondrial DNA. There are only 13 proteins that are produced by the mitochondrial DNA, and most of them are important for the components of the respiratory system of the mitochondria. So what is very important is that the mitochondrial proteins are grouped in different functional categories. So we have a group of proteins that are responsible to import the proteins that are made in the cytoplasm. They are transporters that are able to move the proteins from the cytoplasm into the different compartments of mitochondria and process them. We have the respiratory complex, which is involved in the production of energy in the form of the molecule of ATP. We also have an important group of proteins that are involved in mitochondrial quality control. And there are a few categories of proteins that are involved in this process. Some of them are involved in mitochondrial fission and fusion because mitochondria are very dynamic organelles. They can become elongated or they can shorten and they can exchange materials with each other. There is a group of proteins that are involved in what we call molecular quality control. They are called proteases because they chop other proteins. And these are these proteins that I have here as AAA, LON, and CLPX. And they are important because they can cleave proteins that have not folded properly in the mitochondrial environment. And then there are other proteins that are involved in clearing damaged mitochondria. And the key proteins in this process are these two that I have here, pink and parkin. And the process of removal of damaged mitochondria is called mitophagy. So in normal cells, mitochondria have numerous functions. So the key function for which they are known, and that's why they are sometimes called the power plant of the cell, is the production of energy. But they have numerous other important roles in the cell. And then they have very important functions in metabolism, in the metabolism of glucose, of fatty acid, and this metabolism impacts the energy production. They are also important in regulating ion homeostasis, calcium, iron, and they are also important in apoptotic cell death, which is the programmed cell death, a mechanism that removes damaged cells when they cannot be repaired anymore. So we can see that it has a very important range of functions. And when mitochondria is dysfunctional, that results in altered energy, in energy stress, enhancement of oxidative species, and calcium overload, and cell injury and death. And that has been seen in numerous diseases, including neurodegeneration, cancer, diabetes, cardiovascular conditions. So how mitochondria functions is important in all the tissues, but particularly in the tissues that have high energetic demand, like the brain, the muscle. You can see that when the mitochondria is not functioning, then there are health problems. So what happens when mitochondria is under stress? So mitochondria is under stress continuously from its own respiratory activity to produce energy. So it needs oxygen to produce the molecule of energy, ATP. And this oxygen in the process of respiration induces oxidative stress. So mitochondria is all the time under stress. But there are quality control mechanisms to deal with that. And in terms of protein stress, when there is accumulation of unfolded proteins in the mitochondria, there is the process of molecular quality control, in which mitochondria is able to transmit to the nucleus that it needs more proteins that are helping with the folding or the cleavage of unfolded proteins to deal with this load of unfolded proteins. So this is called molecular quality control. When the mitochondria is not able to deal with stress just through this step, then the second level of quality control is the organellar quality control, and mitochondria is able to undertake processes of fusion and fission to separate damaged parts of the mitochondria and to target them to mitophagy via the PINK1 Parkin pathway that I have mentioned before. When this doesn't work, then the last step of quality control is apoptotic cell death, where the cells, damaged cells, are removed. So it is very important that most proteins that are dysfunctional in the mitochondria, so a recent study showed that more than 40% of the mitochondrial proteins, when they are dysfunctional, they are associated with disease. And I have made a list here that I will not probably go through in detail, but molecular quality control and protein processing induces a range of diseases, including spinocerebellar ataxia, Perot syndrome, Kodas syndrome, which are kind of rare diseases, but they are also involved in diseases related to age-related neurodegeneration, like Alzheimer's and Parkinson's. Other cellular processes, when they are impaired, they can also lead to mitochondrial diseases, which are also rare diseases. And some of them, like the Krebs cycle defects, they don't even have specific names. They are kind of focused on what protein is deficient, and then they are called fumarase deficiency, succinate deficiency, and so on. So proteins in the mitochondria, when they don't work, they are associated with disease. So let's look now at how mitochondria and the nucleus communicate. What are the stress and quality control signaling mechanisms that are taking place when mitochondria is under stress? So as I mentioned, mitochondria can be under stress from its own activity, but also from external stressors. That leads to enhancement of oxidative species and unfolded protein accumulation, because they make oxidation forms and they don't fold properly. And that triggers what we call a transcriptional response, when another sensor in the cytoplasm is able to sense the damage and to transfer that to the nucleus. And in the mammalian cells, the response, this response is called mitochondrial unfolded protein response. There are several transcription factors that may be involved in this process. Some of them are listed here, but in mammalian cells, mitochondrial unfolded protein response is not fully characterized in terms of what are the key components that are participating in that. However, we know that ATF5 and CHOP are players that are important in MTUPR. Another type of stress response, which appears following mitochondrial dysfunction, is called integrated stress response. And it is called like this because it has been seen after several stress factors can induce this type of response. In particular, in the mitochondrial case, mitochondrial dysfunction is able to induce a series of chemical reactions starting from the mitochondria, which have, as a result, translation of a selected number of genes called ATF4, CHOP, and ATF5. And they are the ones that are involved in this transcriptional response called integrated stress response. And I will get back to this integrated stress response and the discussion of whether it is good to have it or it is not. Another type of stress response that is important for the mitochondria, and this has been only recently characterized and identified in the last few years, is the mitochondrial import stress responses. So defects in how the nucleus encodes proteins or how the proteins are produced in the cytoplasm can lead to unfolded proteins and precursor accumulation. If they are not imported properly in the mitochondria, then they trigger this response that is trying to repair the mitochondrial function, either by reducing the number of proteins that are imported in the mitochondria, or by increasing their cleavage with proteasomal activation, or by a range of transcriptional responses that are time-dependent. And chaperones, trying to refold proteins, are the first that are upregulated. Proteasomal activity is the second that comes to try to reduce the load of unfolded proteins. And then another part of the stress response is really trying to alter the mitochondrial metabolism in a way that is making it able to deal with this stress. At the same time, all these responses reduce the expression of genes that are involved in the respiration activity of the mitochondria, just to reduce the stress, apart from the stress that is already on the mitochondria, to reduce the stress that would come from its own respiratory activity. So these are the key pathways which are actually trying to give mitochondria a break from stress in order to repair its own activity. So when these stress pathways fail and there is accumulation of damaged mitochondria, as I have mentioned, there is a process that is called mitophagy that is trying to remove damaged mitochondria. And the key elements that are involved in this are pink and parkin, and there is what we call pink-parkin mitophagy dependent, and there is a branch of mitophagy that is not dependent on pink and parkin. But the majority of the evidence is, and the studies are oriented towards pink and parkin dependent. Of note is that pink and parkin have been found to be mutated in Parkinson's disease, in a small percentage of Parkinson's disease, like 2% perhaps, which is genetic Parkinson's. And this made a very clear link between mitophagy and neurological function and neurodegeneration. So now we have worked for a number of years on how mitochondrial stress is inducing these stress pathways. And what I wanted to show you here is what is the meaning of the upregulation of a stress response. And I'm going to focus on the integrated stress response, which I have mentioned can be triggered by a variety of stresses in the cells, but also by mitochondrial stress. And some of the work that we have done has looked at a dysfunction of a protein that resides in the intermembrane space of the mitochondria. It is called HtrA2. It is a protease, that means it chops other proteins, particularly unfolded proteins. And in the knockouts, in the knockout mouse, is inducing a very strong phenotype. And you can see here that the mouse is very small, has only a 30-day lifespan, and it has a progressive Parkinsonian motor impairment and loss of neurons in a certain part of the brain, in the midbrain called the striatum. And it has also been found that HtrA2 mutations can be found in some patients of Parkinson's disease, but it has also been associated more with early degenerative phenotypes. Okay. So the question was, is this integrated stress response, upregulation of those genes that I have mentioned, is that a feature of neurodegenerative disease? And we have found ourselves that integrated stress response is activated in Parkinson's disease. And a couple of years ago, a paper has shown that this integrated stress response is also upregulated in Alzheimer's disease. So it is upregulated in cell models, in mice models, and in human samples. So the next question would be, is that a good thing or a bad thing? And in order to address that, so I have shown you here that the integrated stress response has, as one of the transcription factors, this protein called CHOP. So what we have done was to do a double knockout with HtrA2 knockout and CHOP knockout, and we have seen that CHOP knockout, the double knockout, has improvement in the neurodegenerative phenotype. So although the stress responses are activated to protect the cell and the mitochondria from damage, when they are persistently activated, so when they do not go back, which is the case of a knockout, of a permanent mutation, they become toxic and they enhance the neurodegenerative phenotype. So in looking at stress responses, these are the factors that we need to consider. Another part of the work that we have done in looking at stress responses and how they may influence the neurodegenerative processes has looked at the mitochondrial unfolded protein response. And I have mentioned that mitochondrial unfolded protein response is generating a series of events that are trying to rebalance the mitochondrial homeostasis. And the first element of that is this protein that resides in the matrix and is called CLPXP. So this protein, when it's activated, is able to cleave unfolded proteins. The peptides are released from the mitochondria and they trigger this series of stress responses. When this activation of the stress response is mild, we have seen that it's enhancing the quality control of the mitochondria at molecular and organellar level, and it's also neuroprotective. It's inducing mitophagy in cells, but it's also improving mitochondrial phenotypes in a Drosophila model of disease that has a PINK1 mutation. So manipulating these stress responses can be used for neuroprotective strategies. So now let's look at how the DNA damage and repair processes may have an influence on mitochondria homeostasis and neurodegeneration. So just a brief background on that. So genotoxic agents, chemotherapeutics, irradiation, ROS, the simple fact that we are living is inducing damage of the DNA. But there are repair mechanisms in the cells. So this DNA repair, the DNA is repaired by endogenous processes, but when it is not, then that can lead either to cancer via changing in the mitotic function of the cells, or it can lead, for example, in non-dividing cells in the brain, to neurological disease or neurodegeneration. And a few years ago, very interesting papers came out linking DNA damage to mitochondrial dysfunction and neurodegeneration. And this link has been found in rare diseases which present DNA repair defects and lead to premature aging disorders. And I have some of them listed here, like serodema pigmentosum, cocaine syndrome, ataxia, telangiectasia. But this phenomenon has then been found in neurodegenerative diseases that are associated with aging processes like Alzheimer's and also Parkinson's. So how that happens? DNA damage needs to be repaired. And that needs an enzyme called PARP1 to function. But this enzyme needs to use a cofactor called NAD+. And when NAD is reduced because it's used for DNA repair, it has a series of reactions, which I'm going to show you more detailed in the next slide, that are affecting mitochondrial function. Then they are affecting mitochondrial function by reducing clearance of mitochondria through mitophagy, and that has an impact in neurodegeneration. So basically, the DNA damage is inducing this requirement for the molecule that is also needed by mitochondria. So they compete for nutrients. And I have it here a little bit more clear, but this may raise a few questions between the non-scientists and scientists. So it's a bit scientific, I admit. So PARP1 is the enzyme that needs to repair the DNA, and it's using this NAD molecule. Reducing the level of this molecule is reducing mitochondrial biogenesis, and it's also inducing 3 pathways by which is determining mitochondria not be functioning properly. One of them is that NAD is needed by mitochondria to synthesize subunits of the OXPHOS and to make the respiration process go well. Another pathway is that it changes the mitochondrial potential in the sense that it's increasing that, and that reduces the ability of mitochondria to be removed by PINK1-dependent mitophagy. And the other pathway is by directly reducing the level of a molecule called SIRT1 that is also reducing mitophagy processes. So there is a feedback loop on this process, which helps then going back to the mitochondrial biogenesis and enhances this. So the key element in this dialogue is really this molecule that is NAD+. And it has been seen that supplementation with sources for this molecule can be beneficial for the diseases that we have discussed, in which there is a DNA defect. We have also done a number of studies in the past where we have looked at combined DNA damage and mitochondrial dysfunction. And we have seen that cells that lack PINK1 function, which are not able to be repaired through mitophagy, they are more prone to assimilate damage at the level of the DNA. Another process where the mitochondria and the DNA are both important is what we call a bystander signaling, because mitochondrial, when the nuclear DNA is damaged, it can transmit the information to cells that are in the neighborhood or at a distance, and can influence healthy cells in inducing also DNA damage in those cells. So we have seen that the removal of mitochondrial quality control is modifying the way in which this bystander signaling is taking place. In some situations, the bystander signaling is inducing a minor stress on those distant cells, and that is a positive effect because it's kind of preparing the cells to adapt to higher stress that can come. And this has been also seen at transgenerational level, that inducing mitochondria using a C. elegans worm model, mitochondrial stress in the mother, there is a protective effect at transgenerational level for about 50 generations. And those 50 generations, they present upregulation of this stress response signaling. So in a summary, in modeling mitochondria and genotoxic stress, we know that mitochondria is normally producing metabolites, and that helps the nucleus being able to transcribe, to produce the genes and the proteins that are necessary for general function of the mitochondria. When mitochondria is damaged, there are stress signaling mechanisms that are inducing gene transcription in the nucleus, and they are turning back to try to help the mitochondria function to restore homeostasis. When the DNA damage takes place and there is an alteration of gene transcription, then there are metabolic perturbations that are inducing impaired bioenergetics and mitochondrial dysfunction. And then what happens then when there are both mitochondria and DNA damage situation? There is a competition for metabolic and signaling resources. And it is actually not clear whether such combined situation would allow for signaling mechanisms of stress and quality control to take place. So this is still something to be studied. So in conclusion, the maintenance of cellular homeostasis is coordinated by communication between cellular compartments. And integrity of both mitochondria and DNA quality control mechanisms are important for each other and for how they respond to stress. And that is relevant for chromosome 8p disorder because we know that the DNA is affected. Chromosome 8p appears to have a higher mutation rate. And there are also clinical manifestations that indicate potential mitochondrial homeostasis imbalance. For example, the link with Charcot-Marie-Tooth and hereditary spastic paraplegia. So here we come to the plan of work that we have. So what we plan is to look at these phenotypes, the way in which cells from the 8p heroes behave in respect to mitochondrial function and also in respect to DNA damage and repair. And as Ethan said, some of these cells, they are a bit more easy to follow, especially for mitochondrial function. Neurons have a very different way in which mitochondrial physiology exists. So if we want to look at mitochondria, fibroblasts are an easier model to conclude on specific mechanisms. And we also have, in fibroblasts, have the ability to look at DNA damage and repair in a more easy way. In our experience with the mice model, for example, the HtrA2 mouse that I have shown you. So the mouse was dying in 30 days, and yet the fibroblasts were looking fine. So we have seen the mechanistic details only challenging the mitochondria and the cell with different stressors. So this is what our plan includes, to look at both challenged mitochondria and unchallenged mitochondria, challenged DNA and not challenged DNA. And we have the tools developed for looking at these details. We are also going to look at how the homeostasis of the mitochondria and DNA damage is modified by supplementation of NAD+ drugs, which we have discussed are available and are important in this dual dialogue. And because these cells are communicated, not only mitochondrial nucleus communication, but they have interactions with other cellular compartments, we plan to look at the nucleus, endoplasmic reticulum, lysosomes, and the details of the cells and the morphology by specific imaging methods, and to analyze if there are any changes in those in the normal fibroblasts and chromosome 8p fibroblasts, and how those changes are behaving on application of stress factors. And finish by mentioning my collaborators. So some of the data that I have shown you has come from the work of Marta Dominguez Prieto, who is today in the group and will work on the chromosome 8p. Some of the work, particularly on DNA damage, was with the help of a student in my lab, Rubab Talpur, and collaborators in Romania, Dr. Diana Savu and Mihaela Temelie. Thank you, and I'm happy to take questions.
Thank you so much, Nicoleta. So yeah, I hope everybody was able to absorb whatever they could absorb. And I want to maybe turn to some of the researchers in the crowd first to maybe kick off a little bit of Q&A. I think Hiroi, I'd love to. Okay, you raised your hand. I was going to call on you because I think you think about sort of aneuploidy. Got a wealth of experience from trisomy 21. So thinking about these integrated stress responses and these other stresses, would love to hear maybe your thoughts, and love to maybe you can invite. You have such experience working with communities as well. Maybe your question can invite some of the audience and some of the families, if they're a little bit scared to come in. Maybe you can invite them in, but please be as technical as you need to be if that's where you want to go with it.
Thank you. This was a wonderful talk. So in Down syndrome in 2019, Mauro's group from Baylor showed that the integrated stress response is one of the key players in the deficits in learning and memory in the Down syndrome mouse model. So what they did was they boosted the EIF2B complex to increase protein translation, and they showed that they were able to rescue the long-term memory deficits that they observe in TS65, which is the mouse model for Down syndrome. I'm wondering if you think something similar might be occurring in AP as well.
We don't really know. I think we need first to see if there is evidence for the integrated stress response. So that will be one of the plans, to look at some of the key transcription factors, but just with simple quantitative PCR, and to see whether integrated stress response or mitochondrial stress response or ER stress response are up. And then we will take this forward. In our hands, integrated stress response, it's good if it's short term. If it's a long-term stress, that can contribute to cell death. And perhaps what you're saying, that stimulating back the translation of other proteins, it's really canceling, having as a feedback loop, the integrated stress response, and is reverting the stress mechanism.
I'll add, I actually did speak with Mauro a few times. This is Mauro Castematioli, that some of you may know about. And Ethan and I had had an update call with him recently. He had some of our fibroblasts. He confirmed there was activation.
Mm-hmm. Okay.
The details behind that, not yet. And he was amidst of a very big transfer and leaving academia too from his, he was at UCSF while I was trying to figure this out. So I know Allison Motri's on the call too, and Allison and I talked about ISR activation a little bit, but this is why I think, Nicoleta, we really would really like someone to look into, the levels. This in a little bit more detail. And from the families, I'm sure the question is, then what, right? What does that mean for us practically?
Practically, in theoretical terms, is if we can detect that the ESR activation is positive, then we can think about it that way. But if we see that this is damaging, then there are ways, as we have heard, to revert that. There are drugs that can block stress responses. So I think this is one way to look at it. The other approach is that if we improve the metabolic pathways that we have seen that are challenged, they may also result in reduced stress responses. And we would have to see that, whether NAD supplementation, for example, reduces the level of stress induction.
So I think, I'm sorry, Mina.
Purpose, just for the families, okay? So why shouldn't our kids all take an over-the-counter NAD supplement? Just in case.
I guess I would take, if it's over the counter and there is no evidence that there is any problem with it. Some of these vitamins, perhaps you want to check with the doctor that a specific patient may or may not have supplements of this type. But I think that in case of mitochondrial diseases, they are kind of given as a support together with cocktails of vitamins.
So I can see from the family. So I can see from the family perspective, again, we want to bring the science back to how is this going to translate? And so I think we've got multiple drug repurposing projects in the hopper. So the hope is that each one will be leading to a set of recommendations. Obviously, even just from Nicoleta's talk and even just from first principles thinking, you would imagine, well, some of these OTC, those over-the-counter sort of nutraceuticals or other things that maybe have been branded as senolytics or anti-aging, but there's a whole universe of these molecules out there, and like, yes, you could probably be led astray. And so we don't want you to spend too much time in that zone because it's not really sort of data-based. But if we can have screens like either with simplified cell models or more complicated cell models, as is happening with RareBase. I also forgot to give a shout out to UAB. I think on the call still is Alexandra from UAB. Yes. There is an effort there to use computational approaches to make drug repurposing recommendations. And so we're going to, hopefully on the next call, actually, we'll get an update about how, on the next Research Roundtable, we'll get an update from UAB and Alexandra's team about what kind of, recommendations for over-the-counter or even other drugs that actually you can only get access through prescription. Like, what are recommendations that could be made based on literature searching and artificial intelligence? So we're trying to take a multi-pronged approach to saying there's many ways that you can come up with recommendations for what to repurpose and what to try. And then, of course, there's things that you would try alone versus things you would try in combination. Once you start considering combinations, more than two, it starts to get crazy in terms of how are you testing all of these things in a rational, methodical way. So we want to not deluge you with all of these recommendations, but we want to have these multiple models, some cellular, some complicated cells, some simple cells, some computational, all of which will hopefully produce recommendations, and there'll be a chance that there'll be overlapping recommendations. And I think that's where we'll get particularly excited about prioritizing. But yeah, I think I would love for other researchers, either Allison, you were called out here, or anybody else, or families, again, use the chat. Camila, you definitely, anyone who wants to raise their hand, please come into the conversation here. We've got about 6 minutes here, and then I think we have some overflow time as well. Oh, you're on mute if you wanted to speak.
Sorry, I don't know why my iPad had, but I didn't raise my hand. It was just my, I don't know why.
No worries, just your
Sorry.
iPad raising for you. No worries. Okay. Anyone else here? Again, I think I like what Bina did there in terms of, I can kind of ask the questions and try to prompt the researchers. I think we always want to make sure we've got the balance on the other side, which is, okay, what does all that science speak mean for practical terms? And so I like the fact that we can use this time to kind of go back and forth and have both audiences engage. Okay, we have Faye raising her hand.
Hi, thanks. I really enjoyed your talk. Thanks so much. I'm just wondering if, when you look at the various genetic differences from all the 8P patients, if you think that, the mitochondrial effects will be the same in all of them. I mean, I appreciate the fact that mitochondrial DNA is distinct, but obviously it's interacting with all the other proteins.
I would not think so, but we will have to test that. So we will have 3 lines and to look at them. But I wouldn't think they will all do the same. So I think it is important what kind of DNA mutation is there in the terms of how that impacts on mitochondrial function and response to additional stress. That's my view, but we will have to confirm experimentally.
And we're trying to take all the possible sort of samples available to us and say, okay, what's a set of inversion, duplication, deletion lines where we can, get a sense of, for example, duplications of different sizes, whereas the rest of the arrangement would be similar, and trying to build a data set where you're trying to learn what happens if you vary the length of the duplication, for example. So we can have a set of samples around that. But then we know that there are duplication onlys and deletions only. Do they behave the same? And so we're trying to now organize this the set of cells that we're going to reprogram first, a set of samples first. We want to consider this question of we want to sample the inversion duplication deletion space, but we also want to make sure we're not missing deletions only or duplications only, but we also want to consider that there's going to be a best order to do all these experiments, because it may not be possible to do everything all at once at the same time. But the thinking here is that we don't want to leave any sort of segment of the community behind. But yeah, we have certain thoughts about how we would design these sampler collections of cell lines from the larger set that we have for these first rounds of experiments.
And sorry, just as a follow-up, do you think, will there be any difference in the mitochondria depending on where the cell comes from? Like if it's from the fibroblast, if it's from the blood, if it's from the skin? I'm presuming not. Oh, there will be.
There will be, simply because the mitochondria in the fibroblasts and the mitochondria in the blood and the mitochondria in the brain, in neurons, they have different physiology. I think, so in what sense? So you will see a general trend that is similar, but the particular physiology of some of the mitochondria, for example, we have seen recently very different mitochondria in iPSC-derived neurons versus the dividing cells they came from. So there is a change in the mitochondrial physiology itself.
Once the IPS is differentiated into neuron.
Yeah. But the fact that if you see a stress signaling or a damage in the mitochondria in fibroblasts, you will most likely see it in blood or in brain. But it will come in a different physiological aspect, I would say.
Thank you very much.
Just wanted to call attention to a great question in the chat here about examples of over-the-counter supplements you can look into, and should you be keeping a log when doing these activities? I definitely would say we need to revisit this topic and have more to say. I think, again, we have these different drug repurposing projects that will lead to these lists of recommendations. And so I think it's premature at this stage, other than just based on for sort of first principles thinking and saying, well, yes, there's an aneuploidy here. There should be this mitochondrial dysfunction. You could take these over the counters if they were available, but I don't want us to I don't want to encourage sort of a free for all here of this, because I think we want to be methodical. But also, if you're going to try things, people should be able to try, but they should do it in a way where they record what they do. So it's a learning for themselves and a learning for the rest of the community. So I think as part of what we've been calling sort of track 4, sort of, the health data analytics side of this and collecting data from patients, I think we want to, come back with a strategy here and not just do things ad hoc, but have a strategy for families that at this stage, when there aren't sort of consolidated lists yet, and when it's sort of just, well, there are things available to anybody if they wanted to try, let's at least try to encourage some sort of quantification of this and capturing some of that data. So yeah, let's definitely revisit that question, and maybe we even engage with some of the folks on the call in sort of like a subgroup context. If we start to have a lot of folks interested in this topic, then maybe a good way to carry on momentum between these calls is to have sort of groups of individuals who are focused on particular follow-up or particular a particular task, and then we make sure that the most interested parties are pushing on it. So we're at the bottom of the hour here. I just wanted to let anyone else who had wanted to say anything add anything. Otherwise, Nicoleta, I think you did an amazing job, and I think Whitney, do you want to preview next time? I think we have UAB up next.
Thank you.
Yeah, I don't have much to say about that. They're going to be presenting their work with their platform, which is an AI platform that looks into the biomedical literature to find connections between genes and drugs. So yeah, we look forward to hearing from Alexandra about that.
Alexandra, you want to unmute yourself and just say hi for a sec just so everyone knows.
Yes, hi everyone. My name is Alexandra Fukshinska, and I am the program coordinator at the Precision Medicine Institute at the University of Alabama at Birmingham. And the Institute director, Dr. Matt Might, he's well known for kind of drug repurposing, but we do it through kind of literature-based drug repurposing. So I'm excited to present next month about our work.
Excellent. Thank you so much. Sorry I missed you in the initial go around of intros. I think with that, unless there's any other questions or comments, I think we'll close out today's session. I'll stop the recording here. Again, just request this link to the video from us after the fact.